Good morning, good afternoon, good evening to everyone. A warm welcome to the 5th Consciousness and Reality Colloquium for this academic year. I'm Kunal Mule, an astrophysicist and the co-founder of IMIX, which is the Institute of Mind, Intelligence, and Consciousness Studies. Christoph Koch from the Allen Institute is our co-host, and we have participants today from across various institutes, including Caltech, MIT, Stanford, and also pleased to welcome Cambridge. The CNR Colloquium series seeks to inspire interdisciplinary investigations on a wide variety of topics which encompass mind, cognition, consciousness, and the nature of reality. Some quick logistics before we introduce our colloquium speaker today. There should be a Q&A button at the bottom of your Zoom window. You can post your questions by clicking on that button, and selected questions from that Q&A box will be taken after the talk. And with this, I now request Christoph to please introduce our esteemed speaker, who is Emmanuel Stamatakis.
Thank you very much, Kunal. Hello, everyone. It gives me great pleasure to introduce Emmanuel Stamatakis. His background is computer science, but he just told me he's actually never worked in a department of computer science. He's always been active in medical imaging, and then moved from medical imaging into imaging regular volunteers. In particular, he spent a lot of time working on language and the language networks that are activated when people speak or try to understand language, and then the default mode network. He's focused many studies on its role in language, but then also now progressively over the last 10 years in consciousness, in particular in patients that have disorders of consciousness, like people in coma or in a vegetative state or in a minimal conscious state. In fact, recently a paper he co-authored really burst into public consciousness, as it were, made it to the front page of the New York Times, showing that people who are behavioral unresponsive, i.e. are considered to be unconscious, can actually repeatedly, reliably, voluntarily modulate their brain activity, and so supposedly are not conscious. He's also interested in other states of consciousness, such as what happens during anesthesia and during psychedelics. In fact, his appointment is cross-disciplinary between the Department of Anesthesiology in Cambridge and the neuroscience in the medical school. Emmanuel, please tell us about your research.
Thank you, Christoph. Thank you very much for the quite generous introduction. I shall share my screen and start. So hopefully you're able to see my screen. Hi, hello, everybody. Sincere thanks for the invitation to talk here today. My name is Emmanuel Stamatakis. I lead the Cognition and Consciousness Imaging Group between the Departments of Medicine and Clinical Neurosciences at the University of Cambridge. So I will start today's talk by trying to explain my motivation. How did I get into consciousness studies? I will describe the tools that I use in my work, and I will try to answer three main questions, or rather trying to relate findings, our findings, the findings from the lab I ran, to three main questions: where, why, and how. And I'll go into that in a moment. So my motivation started from two main interests. One of them was the 2006 paper that was published by a team in Cambridge. And in that paper, Adrian Owen and others described how a patient who displayed no signs of communication was able to respond, or a specific part of their brain responded to a specific command. The command was, imagine you're playing tennis. The somatomotor area shown here by the red arrow increased its activity when the patient was in the scanner and was asked to imagine they're playing tennis, and then decreased its activity when they were asked to stop, and then increased its activity again, and so on and so forth for about 5 times, 30 seconds each time. So this was an indication that a person who doesn't appear to have abilities to communicate understood very well what the instruction was and what was happening in their environment. Now, this happened in 2006, and in 2024, we published a major follow-up from that study that contained roughly 350 patients from all over the place, mostly Cambridge, Liège in Belgium, Paris, Harvard, Cornell with Nico Schiff, and all these places. So we collected fMRI scans or EEG scans from 350 or so patients, and we found that 60 out of 241 participants behaved in the same way or a similar way with the patient I described earlier in the first paper. So this for me was a motivation that got me into studies of disorders of consciousness. The second motivation for me was my interest that started 20 years ago or so on a newly discovered network called the default mode network that didn't match anything we had previously seen at the time. And the default mode network correlated strongly with disconnectivity in the brain and disorders of consciousness. So I'll give you an example here. I hope you can see my cursor. If I focus on this posterior medial area, anatomical names are precuneus and posterior cingulate, and we look at the connectivity of the bars represent the connectivity of this area to the rest of the brain, and we see if you're a healthy control, there's a healthy high bar here. If you are a minimally conscious patient, the connectivity decreases. If you then go into, if you're a vegetative state patient or unresponsive wakefulness syndrome patient, as is known these days, they have even less connectivity, and so on and so forth. And this didn't just happen in the posterior part of this default mode network, but throughout the other parts of the brain that it occupied. So medial prefrontal cortex here, temporoparietal junction here, parahippocampal areas, the picture was all the same. So these were my main motivations on getting into consciousness studies. I have one slide where I would like to describe the tools of my work, which is basically the MRI scanner. We acquire what we call functional scans. We segment the brain depending on the study to anatomical areas or voxels or any other units. We extract time series of the bold signal, because during a single session like the one I described earlier, we may take something up to 300 scans, functional scans. So from each one of those, we derive a time series for each part of the brain, and we build these complex relationships encapsulated in this, represented in this matrix, and we compute properties to make inferences about consciousness. On some occasions, I will show pictures that the colorful or colored-in parts of the brain are where the effect I'm describing is happening. On other occasions, I will show matrix pictures like this, which show more or less relationships in the entire brain. So that's more or less the basics. The themes of my work and what I use to answer most of the neuroscience questions of my work pertain to the functional organization of the brain. So properties from the matrices I showed earlier describe in some way or other how the brain is organized and how this organization breaks down. Besides functional imaging, functional MRI, I work with structural MRI, which in a way is the scaffolding, if you want, that functional relationships are built upon. The questions are asked these days are almost exclusively on the neurobiology of consciousness. I work with healthy volunteers who have been administered anesthetic drugs or psychedelics, and I work with patients with disorders of consciousness, as we described them earlier, minimally conscious patients, vegetative state patients. And I also am very interested on whether consciousness can be manipulated or enhanced in the case of the patients. I'm interested in the old-fashioned meaning and definition of the word neuromodulation. In other words, can isolated brainstem nuclei that contain in neurons that are almost exclusively a single type of neurotransmitter, neuromodulator, do they have any effect on cortical networks and what we call consciousness? So I'll start by borrowing a phrase I saw from a Christoph Koch article. When I say neurobiology of consciousness, what I'm really looking for is the physical footprint of consciousness. So are the areas are anterior areas or posterior areas, according to dominant theories of consciousness, more important in the brain? And I will present some results from, as I said earlier, disorders of consciousness and anesthesia. So to begin with, I will talk about, and within the framework of IIT, I will discuss integration of information and segregation of information in the brains of patients and healthy controls. And we know these are two important concepts in the theory of integration information, because too much segregation creates a fragmented system, and we cannot talk about unified experiences, and too much integration results in undifferentiated systems and not specific experiences. So Andrea Lupi, who was both a master's student and a PhD student in the lab, took some data from healthy controls that received an anesthetic called propofol and a group of patients with disorders of consciousness. So this was Andrea's master's thesis work. And in order to make inferences about areas that are important for consciousness, he computed two indices. The first one is called ICC, and you take the data from each person, each healthy person, and you compute how connected each voxel is to the rest of the brain. So the areas in blue here is comparing deep sedation with awake participants, and concludes that the areas shown in blue are where connectivity is lost with sedation. Now, in terms of segregation, the proxy we used for uniqueness in terms of information content is entropy, was entropy on this particular occasion. It was Shannon entropy. And the areas colored in yellow here demonstrate how richer, how you lost richness in information content as you went from awake to deep anesthesia. The concepts are similar here. The areas are more extensive. So in disorders of consciousness patients, and this particular group contains patients with minimally conscious patients and vegetative state patients, and mostly from traumatic brain injury. And you can see that more areas of the patients are disconnected and more areas of their brain, the patient's brains, are lacking in uniqueness and richness information. In other words, have reduced entropy. Now, if you overlap all of this together, the convergence zone is in a little bit in the precuneus and the posterior cingulate. So posterior medial areas in the brain is the common denominator between unconsciousness in healthy people who receive anesthesia and patients with disorders of consciousness. Coincidentally, that same area was found to be altered in terms of connectivity and in terms of cerebral blood flow when participants this is not our experiment, this is an experiment run by Robin Carter-Harris, who is now in UCSF. The same areas found had functional differences when health participants took LSD or when healthy participants took psilocybin. The largest effects are in the posterior medial areas. So
Emmanuel, can I ask you a question? If you go back 2 slides.
Yeah.
I mean, I know this is correlation. This is not a causative technique, but do you think that tiny red area in the precuneus, if you take an unconscious person, either because it's a patient or it's an anesthetized subject, and just stimulate that area, do you think you could wake that person up if that area becomes active?
We are working on that same data right now, Christoph, with some collaborators in Shanghai who have done exactly that. Unfortunately, I don't have the results yet to give you, but it seems, clearly there is a lot of variation in this group of patients, and TBI patients, brain injury patients, always have huge variability. But what I'm presenting here is a common denominator, if you want. This stimulation in this area may not work for every patient, but this seems to be the overarching picture. If you take anesthesia.
The precuneus, so put it differently, do you think that precuneus is a key node in consciousness? Without that, most would not be conscious.
A key node of the default mode network and a key node in consciousness. Absolutely. Yes.
Okay. Thank you.
So what do we know about this posterior medial area? It's involved in memory, autobiographical memory especially. It has many reciprocal connections with the anterior aspect of the DMN. It's disproportionately large in humans compared to non-human primates. It's an association cortex, and it allows integration both external and self-generated information. And it shares many connections with cortical and subcortical regions. Now, Georg Northoff has done in the past some really nice work, not only focusing in the posterior medial areas, but all along the medial axis of the brain. And we can see huge concentrations of dots, mostly here and here. And what these dots are telling us and what these little symbols are telling us, if we compare my emotions to somebody else's emotions, if we compare my face to somebody else's face, if we compare my memories to someone else's memories, all the activity, all the points of interest seem to be in this medial parts of the brain. So we're getting a little notion that perhaps these areas are areas that are associated with this idea of self, of I. They are strongly implicated in autobiographical memory and have strong connections with places like the hippocampus. Diane Liu was a student in the lab and used she didn't work with patients. She worked almost exclusively with health controls, with data that we borrowed from people. In this particular case, we have a binocular rivalry experiment whereby you're shown different things on the left and the right eye, and your perception of them changes. So in some instances you will see one, in other instances you will see the other. Now, what Diane wrote a very long and heavy and clever methodological paper, which was published in 22. And what Diane found was that the interaction between roughly the areas we're talking earlier, the precuneus and V1, where this was, I forgot to say, this was an EEG, an fMRI simultaneous acquisition experiment. So it offered a rich temporal resolution with the EEG. So Diane found that split the data into states and found that the state that predicts a perceptual transition was characterized by phase coupling between the precuneus and V1. So not only that, but a Granger causality analysis correlated with the perceptual fluctuation rate. So another case and another vote, if you want, for the importance of that posterior medial area for consciousness. Now, after Diane left the lab, she went to Stanford and works with Joseph Parvisi, who does a lot of fantastic work using ECOG. So causality is not longer an issue of mathematics or statistics. So they published one of the best papers for me for the past 3 years that I've seen in the context of consciousness. So these are pre-surgical patients who have electrodes on the brain, and they stimulated in the sides signifying by these dots here. Now, what they found is that in areas that are hot, they are red, they found that a patient's subjective sense of self changed. So they started having out-of-body experiences, or the experiences change in their body ownership. And Diane, given her other work, came up with a working hypothesis that going all the way back to William James, saying that the notion separating the narrative self from the bodily self, establishing that the areas in the anterior precuneus are perhaps more related to the notion of the bodily self, while the other area roughly is the idea, and given its importance for memory and so on and so forth, is more associated with the notion of James and me. Now, can we change this relationship in any way? Can we change if we see these disconnections, if we see these decreases in connectivity with anesthesia or in disorders of consciousness? Can we use some neuromodulator to change this relationship? So we repeated what I have shown you so far, but before I go there, I need to tell you that it was this wonderful study. It focused on the brainstem. We have patients on the right-hand side, where my cursor is, who have lesions in the ventral brainstem, and we have on the left-hand side lesions in the dorsal brainstem, with an area roughly coinciding with what is known as the ascending reticular activating system. And the patients on the left, the patients had lesions that resulted in coma, while the patients on the right did not have lesions that resulted in coma. Now, this was interesting. I'll go back one slide, because we had this finding in our anesthesia study, which related this area in the dorsal brainstem related functionally to the posterior medial area that I have been talking about so far. And it changed its connectivity in a way that correlated with the amount of blood concentration of propofol. So that part of the brain I called the ARAS earlier on, the ascending reticular activating system. is densely populated by nuclei such as the dorsal raphe, uniquely serotonergic, the ventral tegmental area, there's the pedunculopontine nucleus, noradrenaline, and so on and so forth. So we tested all of those to see, in the same manner that I described earlier, whether there are overlapping disconnections between one of them and the brain, the entire brain. So our search area was the entire brain. Our starting area was the brainstem. So we found that the only nucleus in the Aras area of the brainstem that related to both anesthesia and disorders of consciousness was the VTA, the ventral tegmental area, which is purely dopaminergic, and furthermore, its connection that with anesthesia was abolished and reestablished when anesthesia was withdrawn was with the posterior medial part of the brain, starting in the PCC, in the posterior cingulate, and going all the way up to the precuneus. So the finding then, the overall findings, we did an average view on here. They are very tiny. They're shown with this red arrow, but those areas where the overlapping areas where disconnection happened when healthy people received anesthesia and this in disorders of consciousness patients. And that those areas were disconnected with a purely dopaminergic nucleus in the dorsal brainstem. And this started offering a tentative mechanistic explanation why some dopaminergic drugs have been shown to work in disorders of consciousness. Now, again, to mirror the question that Christoph asked me earlier, do I believe that this is the picture in every single disorders of consciousness patients? No, there's huge variability, but this seems to be the overarching picture. What we're doing next is trying to build individual profiles for each patient. And see whether the drug that may work best for them would be dopaminergic or serotonergic and so on and so forth. So I have talked about various experiments, and I know all these areas are not identical, but I think you will allow me to say that they're in a very near each other in a similar part of the brain. And although it's not, this is a picture from a Christoph Koch article, and although they are not extending to the temporal lobe and the occipital lobe, they are exclusively in the parietal lobe, and they are areas that are heteromodal. There's a lot of integration. So we propose, and again, I know that our claim is not identical with IIT, but we propose that those are really critical areas for maintaining consciousness. So I'm moving next to my how question. How does consciousness arise in the brain? And to answer this question, we went back to something that Robin Carhart-Harris, who has done lots of studies with psychedelics in London, initially at Imperial and now at UCSF, has discussed quite a lot. But he's not the only one. And of course, complexity and integration are a great part of IIT and other theories of consciousness. But we focused on entropy and complexity. So we took our data sets to see whether this kind of continuum makes any sense in the context of our patients and in the context of healthy controls receiving anesthesia. So the theory here is that there's a critical zone where humans are conscious. With reduced consciousness, you enter coma, anesthesia, sedation. That's when reduced complexity and entropy is observed. At the other end, you have psychedelic states, you have REM sleep, psychosis, and so on and so forth. So Ioannis Papas, who was a PhD student in the lab, is now at USC, has his own lab, was the first one to observe, to work on this, and compute the entropy of all different groupings of networks in the brain. This is an anesthesia study. So we have awake before this line. To the right-hand side of this line, we have a mild sedation, moderate sedation. People fall asleep, but can be aroused with loud shouting if you want. And we have a return to an awake state. Now, Ioannis noticed that the biggest drop in a network's connection entropy, so the connectivity entropy, the connectivity pattern entropy, was in the default mode network, which includes the posterior medial areas that we have discussing so far. Now, other theories of consciousness emphasize the importance of frontal areas or frontal networks, and here that we see that this specific pattern was not as prominent as the one on the default mode network, and you can see what happened here: recovery, restoration of connection entropy. Thomas Varley decided to look at the similar question, but this time using network fractal dimension. So we know criticality is usually, forgive me if I'm getting this wrong, I'm not a fractal scientist, but criticality is usually associated with higher fractal dimension. So Thomas applied this to awake, healthy volunteers, minimally conscious patients, and vegetative state patients, and you see as the fractal dimension decreased as more unconscious the patient was. And this was on functional data. And Andrea took the structural data and computed the fractal dimension of the brain, a picture of the brain, more or less, not what the brain does, but a static picture of the brain. And indeed, Andrea found the same findings, that fractal dimension of a damaged brain from brain injury can tell you, can differentiate between minimally conscious and unresponsive wakefulness syndrome. UWS and VS are the same thing, but named on different eras. If we do the opposite, if we look at psychedelics, for example, LSD, and this is data we haven't acquired psychedelic data, but Robin Carhart-Harris and others have shared their data with us to sort of check whether our computations work. And these are for the entire brain that I'm talking about now. We see that the fractal dimension of the network increases as we compare healthy controls on placebo and healthy controls on LSD. So we've seen exactly the opposite effect from what we were seeing earlier. The same is noted with healthy controls who have taken psilocybin. However, not as strong as in the case of LSD. Andrea took this one step further, and he used algorithms that are based on a modularity of the matrices that I showed earlier, and separated highly modular states, which are called segregated states, and not so highly modular states, which are called integrated states. And he computed differences, and he found that in the case of LSD, you have an issue during segregated states. So the problem with LSD is, and the phenomena that follow its administration, perhaps some people will find the word problem unsuitable here, but in any case, is during the brain seems to be unable to do segregation as well as when you're on placebo. So although the way you integrate information doesn't seem to change by much, is when it's the case with LSD is when segregation is important. Now, There are a lot of ways to quantify complexity. They're sort of detailed in the lab. Thomas did a fantastic job, and all this job was done during 1 year. He did his master's degree here. He then moved back to the States to work with Ola's Fons on similar topics. But Thomas found that whichever way you use to compute complexity, whether it's sample entropy or Hess exponent or a PCA or as a graph complexity, there seems to be a strong relationship. And here he's using 2 different propofol data sets. So this one, if I remember correctly, is from Cambridge. This one is from London, Ontario. And they both show a similar picture. All these measures of complexity correlate with each other. And furthermore, if you do a component analysis and take a first component, it correlates with a serum propofol concentration. So complexity then tells us something about the state of consciousness. Peter Coppola was another student in the lab who was very interested in consciousness. He looked both at disorders of consciousness and propofol anesthetized healthy controls. Peter was very, very interested in establishing whether the cortex or subcortical areas or the cerebellum was more important or had more differentiating power when you looked at healthy awake people, sedated healthy people, minimally conscious and unresponsive wakefulness syndrome patients. And he found that, first of all, this was the highest discriminating ability between this group. So subcortical areas like striatum, the thalamus seem to be able to tell us minimally conscious from unresponsive wakefulness syndrome patients and from sedating patients. The cortex came second, and the cerebellum third in terms of discriminating power in being able to tell whether these are different from each other. The cerebellum was quite good in telling us minimally conscious from unresponsive wakefulness syndrome patients. What I didn't tell you was that Peter computed the temporal variability of what we call small-walledness. Small-walledness is a network property computed from the matrices I demonstrated earlier. And it represents a situation where you have most nodes in the brain are not directly connected, but any node can be reached from any other. And there's high local clustering and short path length, so global efficiency. So Peter computed how this small-walledness changes in time and then computed its discriminated power in terms of disorders of consciousness. Now, by no means have I got any strong data for the next question, the why does consciousness exist? But I thought I'll give you some data that perhaps talk to that point to an extent. So I'm going to show you some cross-species data. And this is work that Andrea Lupi has done and is doing. Andrea is fascinated by the ability of the brain and his main drive to bring information together. And here for this paper, he went back to principles, information theory principles, specifically redundancy and synergy. It was a starting point we're trying to make it as simple as possible. What do we mean with these terms? So if you take two time series, synergy is the ability, if you want, to see a new signature that cannot be explained by any of the original two time series, while redundancy is the opposite. The information, you can still tell the information as originated from two time series. So in a pairwise fashion, Andrea computed brains, parts of the brain that were more synergistic, if you want, and more more prone to carry redundant information, implying robustness in this case, but also an ability to create information units that sum up more than the individual parts. So red areas here in the lateral surface of the brain, and this is the medial and part of the brain. And as you know by now, I'm particularly fond of this posterior medial area. So red areas are highly synergistic areas. So they're areas that are bring information together. I should also say that this has nothing to do with patients. These are healthy controls. And what by using the neuroscience simulation, he also found that red areas are associated more with more complex cognitive functioning, things like social cognition, cognitive control, autobiographical memory, and so on and so forth. On the other hand, blue areas seem to be coinciding mostly with sensory areas. So robustness of information that seems to be more important in sensory areas. This was not the case in the visual area. Synergy seemed to be quite important in the visual area. Now, I kind of semi-promised a cross-species comparison. We got functional data from macaque, and Andrea found that if you look in terms of redundancy, the information that is carried by the macaque brain is not different from information that is carried by humans. It's not significantly different. However, the ability of the human brain to bring information, to combine information together, seems to be significantly higher in the humans than in the macaques. Now, this is also interesting because this happened to coincide where evolutionary, in terms of evolution, the human brain enjoyed most expansion in the posterior medial areas, that is, and those are areas that are rich in brain gene expression and are associated with intelligence. Now, the same kind of principles I've just described, Andrea went back to the disorders of consciousness, anaesthesia data, and he found that in the posterior medial areas, but also in the anterior medial areas, there is huge loss of ability of the brain to bring information together. And again, this is an overlap between anaesthesia and disorders of consciousness. This is a study, and this is my last data slide for today that Andrea did and looked at anesthesia with sevoflurane in both macaques. Sorry, we didn't do this study. We borrowed data in the kind of climate of open sharing data and open data. And what he did was to compute deviation. So compare the two functional landscapes, if you want, between macaques and humans. So both were undergoing anesthesia with sevoflurane. So you can see how severe the anesthesia was. We have burst suppression here. And so what we found was that the macaque brain, even if it didn't have the anesthetic, human brains, when compared to macaque brains, even if they don't have anesthesia, are more similar. So the human anesthetized brain becomes more like a macaque awake brain when it receives anesthesia. And the pattern is also similar when macaques have anesthesia. So again, I mumble my words a little bit. Here's comparing anesthetized human brain to awake macaques, and here's comparing anesthetized human brain to anesthetized macaques. And you can see the restoration after anesthesia ceases to exist. And the plots show distance, Euclidean distance between the two functional patterns. So one last slide is not our data. It's they are summary papers actually, and they trying to address one of the ideas that consciousness may have evolved to help us understand others' minds. And the default mode network, these posterior medial and anterior medial areas that have been talked throughout this talk, are the ones, but also actually the angular gyrus, which is also part in the lateral surface of the brain, are areas that are strongly implicated in this social cognition ability that we have. So I've given you a lot of data from a lot of different sources. I hope I have managed to persuade you a little bit. This is not a picture I have drawn, but got from this paper, that phenomenal consciousness has something to do with the posterior medial parts of the brain, that this can be influenced by the brainstem, especially by the ARAS system and the thalamus. We have some data to support that on the thalamus, but I didn't have time to show that today. And attentional networks and frontal cortices have a role to play in consciousness, but it's not as important as the posterior medial areas in consciousness. So you've seen these people pop up throughout the presentation. They have done a lot of the work. If you're interested in consciousness, I expect you are, please come and join us for the 28th annual meeting of the Association for the Scientific Study of Consciousness. It's happening in the town I was born in, Crete, and I'm co-chairing it with Athena de Merzi from Liège. This is the lab link that will take you to an up-to-date publications page. You can link to all the papers I talked about today. Thank you very much for your attention.
Thank you very much, Emmanuel, for that excellent talk. While the questions are flowing in, and I know Christoph might have a couple of quick comments or so, but let me quickly ask you that given your excellent studies on human consciousness that you presented, taking it a step further, what are your views on consciousness in general? What is consciousness, and is it limited in species having a brain or neurons? And taking it a little further, what is reality or different forms of reality, let's say in altered states of consciousness?
I would, to specifically address your question in a sort of kind of reductionist way, I think consciousness is what allows us to interact with our environment on a daily basis. It's based on the model that already exists in the brain, and it's influenced by that. I believe, although I haven't read or studied a lot on this topic, or panpsychism, which is, I think, where you're trying to go, that yes, neurons are probably necessary for consciousness. can't remember.
How would we know?
Another angle to your question.
How would we know, Emmanuel, if it's...
Well, that's a fair question.
That's a problem.
Christoph, but that's a belief rather than a scientific finding.
It's fair enough. Christoph, do you have any quick comment, question? Then we'll go into the Q&A.
Emmanuel, you really point out and I get it, and I think the data is very strong, the importance of the posterior precuneus. But let's say if I hear something, or I see something, or I feel something on my skin, okay, so sensory consciousness. What do you think is active there? Is it the precuneus plus visual cortex, or plus auditory cortex, or plus somatosensory cortex, or what?
Absolutely. I have no doubt about that. There will be difference on individuals, how they process this sensory stimuli and how they reach higher cortices. And I think those differences will be based on, if you want, their experience, their priors, if I want to dip my toes into another consciousness model. But yes, they absolutely, primary sensory cortices are important. And they are important for consciousness. I think that if you remove the ability of a person to hear or to see, they have, and if we accept a continuum of consciousness, they have probably a slightly different state of consciousness from somebody who can see or can hear. I'm not making, I should clarify that this is not a value judgment, it's just a different thing. Yes, absolutely. Sensory cortices are important. I believe the whole of the brain is important. And I think the pictures that I showed today perhaps tell a story, which is these are more important in a way that is defined by statistical thresholds, if you want. But
okay, but concretely, if I have a hole in my prefrontal cortex, let's say I lose most of my prefrontal cortex, everything in front of my premotor cortex.
Yeah.
I might not be able to speak, but do you think I would still be capable of experiencing sounds or sights or smells or touches?
Absolutely, yes.
Okay.
I think it will be more difficult to have a subjective experience if the posterior medial part of the more posterior part of your brain has the same fate. Yeah.
Okay.
All right, excellent. Let's quickly go on to the Q&A. Some questions posted by the participants. Here's an interesting one asked by Siddharth, who says, the way you have presented consciousness of the brain regions, how early in one's life does this kind of consciousness develop? In the fetus itself, or also how far in terms of animals or insects can we take this kind of consciousness?
So, I mean, towards the end of my discussion, I talked a little bit about macaques and primates. So I have no doubt that there is a level of consciousness that is different from a human consciousness. And so far, what we have found is that the difference is that the human brain is able to make more out of bringing information together. I'm so sorry, I forgot the beginning of your question.
How early in one's life does this consciousness?
Oh, yes. So there is increasing evidence from neonatal studies. And it shows that the default mode network increases its connectivity, strengthens its connectivity, and that includes the posterior medial areas that I've talked about. As we develop in life up to something like 60 or 70, where it kind of stabilizes. And of course, its relationship is also with medial temporal areas, the hippocampus or the medial temporal lobes, which are all important for autobiographical memories. I think all those are related, and yeah, that's how in developmental terms, this connectivity of that specific network changes across the lifespan.
Excellent. Here's one by Divya Chander from Stanford, I believe. We tend to find in anesthesia and other disorders of consciousness or conscious states that the DA to ACH balance is more important than just ACH or DA alone. How would you reconcile that with some of the posterior medial nuclei you have been observing disconnected that are conserved during anesthesia and disorders of consciousness?
Allow me to read the question. I have the question in front of me, and I want to understand it a little bit more. So we have the dopamine acetylcholine balance more important than just, I mean, that's, I firmly believe that if you, all those are interconnected, and if you change the one, you'll have some sort of consequence in the other. In data that we have, I mean, and by no means did I suggest that dopamine is the only thing that modulates the brainstem posterior medial relationship, functional connectivity relationship. In fact, we have an analysis that takes into that with LSD and psilocybin, and it shows that the median raphe and the dorsal raphe are the predominant forces and the predominant areas of connectivity change. Again, with posterior medial areas, we do not make a targeted connectivity analysis. We consider the entire brain, but the raphe nuclei exclusively serotonergic. The relationship with posterior medial areas changes after you've taken LSD and to a lesser extent after you've taken psilocybin. So to answer your question, Divya, I don't think, I think as a scientist, we redact, we take an average, an overarching picture to make an initial conclusion that hopefully will act as a springboard for further exploration. And those relationships are a lot more complicated than I had time to present or I have indeed done research for.
Thank you. Here's one by Nancy Hopkins from MIT, of course. She asks, have you or anyone else studied transient global amnesia, which is a sudden temporary loss of memory where individuals cannot recall recent events or form new memories for a period of time, which is typically up to 24 hours?
I very sorry, Nancy, I don't know of any, I haven't, and I don't know of any studies, but the field of consciousness is increasingly enjoying participation, and I want to be part of this from scientists who study dementias, all kinds of dementias. So if there aren't any studies, I sincerely hope we'll see some studies on the topic really soon.
Thank you very much. I think we're out of time, so we will end here. Emmanuel, thank you very much for that excellent talk again. So this was the final colloquium for the current academic year. The next lecture will be the inaugural colloquium for the next academic year, which is scheduled for the 29th of October, and that will be delivered by David Chalmers from NYU. So stay tuned for more information on that. And finally, again, thanks, Emmanuel, for being here, giving such a thought-provoking talk. Thanks to our co-host, Christoph Koch, all organizers and participants who made this event happen. Have a great summer break, everyone.
Thank you very much. Bye-bye.
Thanks again.